DNA Replication by Sammy and Judah. Before we begin, it is necessary to understand the cell cycle and which steps of DNA replication will be occurring. The first stage of the cell cycle is gap 1. Synthesis comes next, then gap 2, and finally mitosis. For this video, we will be looking only at DNA replication in synthesis. Here is DNA, made up of nucleotides which each include a phosphate group, a deoxyribose sugar, and a nitrogenous base. Now, let's meet the cast, or the important enzymes used in DNA replication. The first enzyme is helicase. Helicase unzips or separates the two strands of DNA by breaking the hydrogen bonds between their bases. The next enzyme is primase. Primase lays down RNA primer, which is used to direct polymerase on where to build. Polymerase, the third enzyme, builds new strands of DNA by reading the primase 3' to 5' and building in the 5' to 3' direction. We'll go over 3' and 5' later. The final enzyme is ligase, which fixes Okazaki fragments and glues together uneven DNA strands. Going back to 5' and 3', these classifications are determined by the deoxyribose sugars on each strand. The strands are labeled 3' to 5' because of the carbon orientation of the deoxyribose sugars. These directions affect the way that the enzymes will travel through each strand. The first step of DNA replication is conducted by helicase, which runs through the hydrogen bonds between the nitrogenous bases and opens up or separates the strands. The second step is conducted by primase, which lays down RNA primer in the 3' to 5' direction. The third step is conducted by the polymerase enzyme, which reads the primase 3' to 5' and lays the daughter strands 5' to 3'. The fourth and final step is conducted by the ligase, which will fix the Okazaki fragments and glue together uneven DNA strands. Our DNA has now been replicated. Each new DNA consists of one parent strand and one daughter strand. It is important to note that the parent and daughter strands both have 5' and 3' classifications. Thanks for watching.